All right, I don't know where everyone is, which is sad because this is really like interesting and exciting to talk about. But you know what? We're just gonna go with it. Remember that you have the primary lit assignment due on Friday. It is posted. Some of you guys have already emailed that to me, and if you have other questions about it, let me know. So as you can see, today we are talking about aspects of breaking tolerance and autoimmunity. There's a little bit of a preamble I have to tell you before I get into some of the information that I'm going to tell you here about breaking tolerance and autoimmunity. And the preamble kind of has to do with some of the next places we're going in this semester. So when I look at sort of the next few topics we're going to be talking about on the syllabus, you can see that one of the things that we're talking about is autoimmunity, which is up on the top right of this slide. You can also see that the topic after autoimmunity that we're going to talk about is called hypersensitivity, which, spoiler alert, is the immunologist's word for allergy. So you can see allergy is up here. And then after hypersensitivity, the next thing we're going to talk about is transplantation. And what this sort of slide is getting at, and what I want to sort of mention a little bit as a preamble today, is that in a lot of ways, for immunologists, these three topics have a lot of conceptual overlap. So there's a lot of similarity between these three topics. In fact, this table from an older version of your textbook talks about how you can actually think about different mechanisms and see similar mechanisms happening between these three phenomena. So what I want to kind of mention with regard to all of this is that if we're being super technical in terms of immunology, if an immunologist says autoimmunity, they basically mean an immune response to self. If an immunologist is talking about transplantation, it's immune response to other people's organs. Or other people's tissues. And if we're talking about hypersensitivity, we're thinking about immune response to something that's foreign but harmless. Sometimes you'll see in textbooks people write about the innocuous antigen. And I don't know how to spell innocuous. And so one thing to note is that sort of, these three things have in common, and the way that I and many immunologists think about these three things in common is kind of related to Goldilocks. These are the examples of too much. These are all the examples of too much immune system. And in other cases, there's this other side where we see immunodeficiency, which is too little. So again, immunologists often think in terms of Goldilocks. with all of this. And we are generally, like I said, dividing up these kind of too much immunity things based on what the antigen is, whether it's a self-antigen, whether it's some other person's tissue, or whether it's a foreign but harmless thing like, say, pollen. One thing that you can notice just from thinking about that is I'm telling you that we're distinguishing these things based on antigen. And because of that, you can infer that we are going to be seeing important actions of adaptive immune responses, because we often think about a specific antigen if we're thinking about an adaptive immune response. There also are situations where we have too much immune response, but it's too much innate immune response. And those are usually referred to as inflammatory diseases. Or sometimes people talk about autoinflammatory, which is sort of too much innate immune. Response. The reason why I'm telling you this all now is that officially, as you could see from the title slide that I made for today, we're thinking about breaking tolerance and autoimmunity. However, in a few cases of the examples I'm going to be telling you about today, immunologists could argue about whether autoimmunity is really the best term for them. Because sometimes, like when I can't breathe because of pollen, it's really easy to say, yeah, that is a foreign harmless antigen. And when we're thinking about, say, type 1 diabetes, where CD8 T cells are killing pancreatic beta cells, we can say, like, yeah, that is clearly an immune response to self. But there are some other situations where sometimes it's a little bit harder to say, is the problem too much inflammation? Is the problem response to self? How much is the problem is an outside antigen? So some of the situations are a little bit grayer and don't sort of divide as nicely here. And in particular, some of the examples I'm going to tell you about today are diseases that I think people would debate about whether or not they are autoimmune or whether they are autoinflammatory or whether they are hypersensitivity. And so I figured I needed to sort of tell you this all now, just because technically you might see something that we're talking about and be like, is that really autoimmune? And my answer is, yeah, I agree. It's a gray area, but it's a perfect example for the thing I want to talk about, so we're talking about it today. So just be aware of that. The other reason why this is interesting and related to some stuff we're going to see today, well, kind of, maybe today, depending on timing, is that because autoimmunity, transplantation, and hypersensitivity are all similar in that they are sort of too much immune response, too much immunology, they have some similarities in how they're treated. And so because in every case, we're trying to turn down immune responses. And so in some cases, we see some similarities in treatments. So if I so that's actually because of timing, I end up kind of putting the treatment stuff kind of wherever I can fit it, because the same treatments often work for many of these things. So we will see this slide over and over again as we're talking more about allergy and transplantation, and we're thinking about the specific mechanisms. Today we're going to be thinking more specifically, as I said at the beginning, about breaking tolerance and autoimmunity. There's also one other really key thing that is sort of an overview-y thing that comes up today. And this has to do with the fact that more and more often we are realizing that there is a role of infection in autoimmunity. And so in a bunch of my examples that I will show you today, you will see an infection either directly, very obviously being part of what's going on, or kind of like vaguely, we're like, and then there was pathogen around, and there's sort of being an infection there. There are many, many, many hypothesized roles for infection and autoimmunity, but this is something that has been really hard to always prove. And so I will say you're going to oftentimes see some examples, and often today it's going to be like, oh yeah, there's clearly some infection situation going on here. But it's actually really hard to sort of always nail down what's going on. One reason is that sometimes what happens is that there needs to be a particular microbe in a person who has a particular HLA type. And so it's really hard to be like, oh, does Borrelia always cause arthritis or not? It seems like it does in some people and not other people. And unless you happen to actually do HLA typing, you don't figure it out. And you're just kind of like, this is random and weird and I can't figure out what's going on. And so some part of it is like, it's really hard to make the association because we all don't know our HLA types off the top of our head. And so trying to figure out that association between the microbe and the infection, or the microbe and the autoimmune disease, is hard. You can see some examples, and this is from the most recent version of the Parham textbook. It's a bit out of date, of some situations where there are particular microbes that have been associated with autoimmunity. I would say that this is a dramatic underestimate. There's one other, and so there are a bunch of them that are sort of hypothesized in the literature that no one has been able to prove for like years and years and years. And only in more recent years has there really been good data. So for example, there's some fantastic data now relating multiple sclerosis to a particular viral infection. And one of the reasons why they were able to do that is because they actually needed huge numbers to actually deal with the variation in the HLA types. And I don't remember the number off the top of my head. I can go back and look at it. But if I remember correctly, they finally did it because they got access to a online database with information about 8 million people. And so it was only because they could actually get that much data. And I can go into more of the details of the study of that. They were actually finally able to go from sort of like this rumor that had been around for a long time to actually getting some legit data. There is a paper that came out within the past week about another virus and lupus. That again, I think is a similar kind of thing. I will admit I have not yet read said paper, but it got big press. But I'm doing a podcast episode on Friday, so if you ask me, so I will have read it by Friday, and I will know some things about it then. And so, there are becoming a lot more sort of concrete examples of this. We can't always give you the specific concrete example. There's one other reason why actually coming up with a particular association in these cases is really, 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 really hard. So we're going to be talking a lot today about how breaking tolerance works. How it works that we stop having some kind of peripheral tolerance. And as we'll see in these examples, we can imagine roles for infection in a lot of it. But what that means is that in that situation, when you have the salmonella that might lead to reactive arthritis or whatever, what's happening is that you are impacting a B cell or T cell and breaking its ability to have tolerance. That one. You still need that cell to actually interact with the antigen again and do clonal expansion and maybe start to do damage to the body. And sometimes the timing between that tolerance break and when you actually see the symptoms and go to the doctor and are like, there's something wrong with me, can be years. And it's really hard. You might have gotten rid of the infection by then. It might just be that it took a while to add up all the damage that those no longer tolerant cells could do. And so it's unlikely that you're not going to still have that microbe around in your body. And it's unlikely that you're going to be like, yeah, you know, doctor, I had the sniffles one time. That must be why I have this autoimmune disease today. The linkage time-wise is pretty far. It's really hard for you to necessarily make that cause and effect linkage. And so again, this is why some of these newer gigantic studies are really just now giving us some information about this. And so what I will tell you is in a lot of cases, we can say there is very likely an infection that was involved in the tolerance break, but it's often really hard for us to say like which infection, what time, like when that happened. There was probably an infection involved, but the exact details are a little bit unclear. So we can think about breaking a bunch of our different types of tolerance that we talked about. I told you last time that we can't really break deletion. Once the cell's dead, we can't like get a Ouija board and bring it back to life. Similarly, we can't really break helplessness either, because if the B cells can't get help because the T cell's dead, you can't bring the T cell back to life. So those two are sort of harder to break. Much of what I'm going to be talking about today has to do with breaking anergy, suppression, and deviation. Those are sort of the big hitters for this. The next slide does mention one mechanism that you can sort of imagine as being related to breaking ignorance or breaking immune privilege, although it's not like perfect. And this mechanism can actually be involved in a lot of different places. But this one thing, I put it here because it does kind of tie into ignorance, is something known as epitope spreading. Also sometimes it can in fact also relate to some of the other mechanisms that we'll see in terms of amplifying them. So you can see it's also listed here as this autoimmune amplification, because in fact epitope spreading can happen in a lot of the cases we're going to talk about to sort of make things worse. And the basic idea with epitope spreading is that sometimes there might be some antigens. Shown here is the little triangle, this green thing and this red thing, that may be present in a cell and perhaps aren't being presented very much or are maybe not secreted, so B cells can never make antibodies against them or whatever. But if that cell dies and we release some of those molecules, suddenly those previously hidden antigens may become visible to the immune system. And so the idea is that basically if there are cells that die, you might have antigens that were sort of intracellular antigens that were sort of previously hidden now get involved in processes. And so you could imagine this could be something that's happening when something's immune privileged, if we're hiding some antigen. If we suddenly have some cell death or if we suddenly have, the trauma to the eye, we're going to release the antigens that we couldn't see before. We can also imagine, but also there are cases where if we start to attack some cell with one of the other mechanisms we see, sometimes we also release other antigens, and now we don't have a problem with one self-antigen, we have a problem with multiple. So that's sort of this idea of epitope spreading, where, again, we spread from having a problem with one epitope to having a problem with many. And this could amplify many of the things that we're going to talk about. But a lot of the big mechanisms are mechanisms that are related to clonal anergy. And so you can see clonal anergy here. And specifically, we're talking about T cell anergy. So what you can see is this T cell. Of course, when it is a naive T cell, it requires signal 1 and signal 2 in order to be activated and proliferate. So it needs to see MHC plus peptide with its T cell receptor, as well as seeing B7 with CD28. And remember that the only reason why that antigen presenting cell makes B7 is because there's been PRR stimulation. So it's sort of telling the T cell, hey, there was something bad around. You need to be fully activated. You need to actually be ready to do something. And we talked about the fact that if our cell just sees signal 1 without signal 2, that's likely a self-antigen presentation situation because there's no microbe around. And so our T cell is going to become anergic. And that's one of our mechanisms of peripheral tolerance. You can also see that what this shows on the far right is that if that cell later sees signal 1 and signal 2, it's going to stay off. It's still going to be not working. There's one mechanism here. There's one thing that's actually not shown on this slide that I want to just mention. I just double checked how I had my slides set up and realized I didn't have another specific place to talk about it, so I'm going to talk about it here. And I would say that anergic cells are off asterisk. This shows that cells staying off and staying anergic. There are situations where the cell could get turned back on. The cell is not 100% all the time off. It's like 99% off, with a rare chance that it could get turned back on. And that rare chance where it gets turned back on involves it seeing signal 1 and signal 2, like you see here, as well as so many cytokines. Sometimes something that's referred to as cytokine storm. Just like crazy high amounts of cytokine. And you can imagine that if they're crazy high amounts of cytokine, particularly crazy high amounts of inflammatory cytokine, something bad's probably happening, right? What bad thing is probably happening if there's crazy amount of inflammatory cytokine? Yeah, Jazz? A really bad infection, right? You got a really, really bad infection. If you have a really, really, really bad infection, there's a certain amount where it's like, you know what? Let's turn everybody on. We need every T cell to participate. We can't have anybody sitting on the sidelines. This infection's so bad. Basically, it's like our choices are die of this very bad infection, or maybe turn on a problem T cell. But that's not going to kill us today, so that's okay, because we're going to live through the terrible infection. It's okay that we turn on the problem T cells now, because we're in the emergency situation. We got to turn on everything just in case. And so there is some idea that after certain very severe infections, you may actually end up with some tolerance broken, because you may get some of your previously energized cells turned back on. And like I said, I don't have a specific other slide for that. But I will say that if there is a situation, again, you could imagine this would be some infection. But we can't necessarily say what it was. When there's some infection, you might, if there was a T cell that was around the right location of your body, it just happened to be in the wrong place at the wrong time, and get those crazy cytokines, it could get turned on when it shouldn't have been turned on. It can get unenergized. It can get woken back up, and suddenly that could start leading to some autoimmune processes. And so that's one place where you could see that infection leading to autoimmunity that I don't have sort of described anywhere else. So that would be one way that energy could get messed up. But there are a couple of other really big ways that energy could get messed up. The next slide I have listed as mechanism 1. I guess technically this is mechanism 1 and the other one's number 2, but whatever. Let's just say I can't count. And this is one of the ones that freaks everybody out. It's also very clear from this one a way that infection could be resulting in autoimmunity. So we've talked about the idea that we have antigen-presenting cells like this dendritic cell or whatever, presenting self-antigen, right? We're resting. We don't make signal 2. We don't make B7. We interact with that T cell. The T cell gets energized, right? Notice that this antigen-presenting cell, and this is true whether it's a dendritic cell or any other antigen-presenting cell, doesn't have one copy of MHC on its surface. It has many MHCs on its surface. We talk about, the 6 class 1s and the 6 class 2s. But in fact, it's not just like 12 if it's a DC. The cell might have like 100 copies in some cases of that same MHC class 1, of HLA A17 or whatever. This is showing 2 because it's hard to draw 100. They don't all have to have the same peptide in them. So you could have a situation where that dendritic cell is presenting multiple different peptides in different versions of the MHC molecule. Now, if you have an infection with a virus, for example, it's very likely that that infected cell is making a ton of virus proteins, and the majority of the MHC molecules on the surface of that cell are presenting viral peptides. But it's not all of them. It's not 100%. So you can imagine, maybe in the case of viral infection, your antigen-presenting cell is presenting 99 versions of the viral peptide. 99 of the MHCs have the virus stuff in it, and 1 has self still in it. Because the cell isn't going around the cytoplasm like, I want to present this peptide and I want to present this peptide. It's just a math probability thing of which peptides get up there. And so there is some frequency of self peptides that are still getting presented in that infected antigen presenting cell. And so you can see this at the bottom. Here's our antigen presenting cell, gets infected with a microbe. You can see in this case, that antigen-presenting cell is presenting self on some MHC molecules and is presenting foreign antigen on other MHC molecules. Again, here it's 50-50 because they only could draw 2 because drawing's hard. It might only be, 1% versus 99%. But now this antigen-presenting cell has been infected with something. See, it got infected with something. Now this antigen-presenting cell is going to make B7. Because it's going to be making a signal 2. It really needs to turn on this T cell that is responding to the foreign peptide that is being presented. We really want to turn this one on and get rid of the microbe. So we're going to make that B7. But that B7 is a transmembrane protein in the membrane of this antigen-presenting cell. And as we've talked about, proteins in the cell membrane, proteins on the surface of cells, are not sort of statically in the membrane. They don't just stay in one place all the time. They diffuse around and are next to different things at different times. That B7 doesn't know that it's supposed to only be with this MHC plus peptide. That B7 can now also pair with other MHCs that are on the surface of that cell that might be presenting self-peptide. And so because of again, bad luck that this infected cell was still presenting a little bit of self-peptide, it now has the ability to turn on a self-T cell, where that T cell should have been energized, but now it's not. Now that T cell is going to get activated because it's going to see signal 1 and signal 2. Because in this case, the antigen-presenting cell is making signal 2. Now, this is going to be a relatively rare occurrence because most of the time, you are making new T cells 24/7 throughout your life. And usually, those T cells are going to get energized. The first thing they're going to see is going to be self, and they're going to get energized. So imagine like every T cell you've made up until before this point got energized. If you happen that day to have made a naive T cell that has not yet been energized, and it happens to come across its antigen being presented in the context on an infected cell, even though it's infected with something else, if it just so happens that its antigen is the one rare one that got presented and it was not yet energized and it comes across this infected antigen presenting cell, it's going to get turned on where it shouldn't have in other situations. And so now maybe all your other T cells against that antigen are energized, but this one's not, so it's going to proliferate and make a clone army and give you autoimmunity. So again, you can notice there is some role for an infection here, though it doesn't have to necessarily be a specific infection, and it's really hard to kind of tie back to what it is. There's a fair amount of bad luck happening in this scenario that results in this T cell getting inappropriately activated. But this is one way that we can see energy get broken. The other really big way that we can see energy get broken, and also people always want to have like a name for this one, because the other one I'm going to tell you has a name. So they're always like, well, what's the name for this one? And I'm like, I don't know, like inappropriate B7 sharing. Like I don't have a name for this one like I have a name for the other one. But you can make up whatever name you want for it. But the second one has a name. And the second one is called molecular mimicry. And so the idea with molecular mimicry is that there can be situations where a peptide from a microbe is very similar to a peptide from self. They might be identical if we're thinking about some of the particularly class 1 epitopes where we're only talking 8 to 10 amino acids long. So probability-wise, there could be a situation where there is an 8 to 10 amino acid long peptide in the pathogen and in a self protein that are the same. Or we can see a situation where they're not identical, but maybe they have parts that are the same. So here it shows just that the like tip part is the same and the rest of it's different. The idea is that they look similar. Exactly how identical they have to be is, debated for different times. What that will mean is that when we are infected with this pathogen, we will activate naive T cells appropriately because we want to get rid of the pathogen. Like, yay, we want to get rid of the pathogen. We're going to attack with these T cells, as we should. But now we've activated these T cells. These T cells are no longer naive T cells. They are now effector T cells. Now they don't have that constraint on them anymore of needing signal 2. And so now if they see that basically exact same peptide or a really, really similar peptide being presented on a self cell, even though there's no infection around, there's no B7 around, we're going to be able to see action against that self peptide. We're going to now be able to see this molecular mimic, this similar self peptide be attacked after the T cell was first activated by a foreign peptide. One thing to note here is that this one is much more HLA restricted. So usually this is why we might see, that infection leads to autoimmunity only in people with this HLA type, because only that HLA type presents that particular pathogen peptide and that particular self-mimic. So if you have the bad luck to have that infection and that MHC type, you could inappropriately have these cells get turned on through molecular mimicry. But everybody else who has that pathogen doesn't have to worry about it because they can't present the mimic peptide. Everybody who has that MHC type who doesn't get that pathogen doesn't have to worry about it. It's only in sort of that I have the wrong MHC and I got the wrong pathogen. And suddenly through molecular mimicry, I'm getting this activation. Yeah,
so
Paige.
It's not going to be activated from the
So the mimic self-peptide is usually going to be shown without signal 2. It's only going to be with signal 1. So the T cell, the naive T cell in that case, isn't going to be able to get activated here because the self-peptide isn't presented in a context with B7. But once it's been activated by the pathogen, now it doesn't need that second signal anymore. Okay? In a lot of ways, molecular mimicry is really just sort of another way of thinking about cross-reactivity, which I told you about before, sort of this question of, are the peptides similar between pathogens or between pathogens and self? You know, there's only 20 amino acids. There's only so many choices we can have for different types of protein structures. And so this is sort of another version of sort of what this looks like, is that we can imagine that perhaps we have a situation where someone's infected with some virus that turns on a particular CD4 helper T cell subset, a particular CTL subset. When that CTL subset leaves, it's going to kill anything that has the peptide of interest on it. And that could be things that have the virus. But in fact, by chance, that also could be the beta cell in the pancreas that just happens to have that self-peptide being presented. And that can lead to killing of all of the beta cells in the pancreas and type 1 diabetes. In fact, that kind of mimicry is what we really think probably happens in type 1 diabetes, is that the CD8 T cell is getting activated by some molecular mimic viral protein, though of course it could be one of the other mechanisms of breaking tolerance as well. One other interesting thing that we sometimes will see is that people have actually done experiments and they've looked at situations where the T cell receptor binds to MHC plus peptide. And on the left, you can see the blue. Well, in all of them, the blue is the MHC and the little green is the peptide. And then the red and the yellow are the two TCR chains, alpha and beta. And this version that you see all the way on the left is basically a T cell receptor recognizing an influenza peptide. This is like what it's supposed to look like when it's a foreign antigen. And we've even seen that sometimes if the T cell comes at MHC plus peptide at the wrong angle, it can look like a mimic peptide. It can look like the wrong peptide and the T cell can get activated because of the geometry is actually wrong. And so it looks like a different peptide than sort of what the T cell should be normally recognizing. And so here's an example of one off-angle recognition. Here's an example of another pretty dramatic off-angle recognition. Both of these are examples coming from multiple sclerosis patients. Because of obvious reasons, I've been talking a lot about T cells in thinking about all of these things, but I will absolutely say that antibody responses can also show some of these same situations. One really, really, really famous example is something called rheumatic fever. So in rheumatic fever, people get infected with a particular type of streptococcus, which has a particular molecule in its cell wall that we induce antibody responses to. You only actually induce these antibody responses if you have a particular HLA type, because you need the right kind of T cell help. But if you make that particular antibody response to that molecule, it turns out that molecule is also found in the heart. And antibodies float through the blood once they're secreted from B cells. So in fact, they're going to eventually bind to the heart and cause this disease that has a bunch of damage of heart tissue called rheumatic fever. And so the point that I want to make here is that this is another example of a cross-reactive antigen. It's just now we have an antibody doing the cross-reacting and not a T cell. And what I want you to see is that, yeah, this molecular mimicry issue, this cross-reactivity issue, is not just a T cell problem. It could also be an antibody problem. I just like talking about T cells more than I like talking about B cells. So you can see this example here. So at this point, I'm guessing, or I'm hoping you notice the importance of bad luck, unfortunately, in a lot of these situations. The other really big set of mechanisms that we think about when we think about mechanisms for breaking peripheral tolerance are mechanisms that have to do with clonal deviation and clonal suppression. In a lot of ways, these mechanisms are all kind of linked together, and so I'm not going to separate them into deviation or suppression. Because if you remember, deviation was that you kind of made a different type of helper T cell subset. So in this case, Th2 cells were protective while Th1 cells caused damage. And so the idea is if you are fortunate enough to make a Th2 response in this case, you're going to be okay. If you are unfortunate enough to make a Th1 response, you're going to get some damage. And we could argue where that damage falls in my little scheme that I wrote on the board. No one cares. This is showing this, and this is and when we think about deviation, a lot of times we kind of talk about Th1 versus Th2, or sometimes we'll talk about Th2 versus Th17 or something like that. But recall that when I drew out all the different sort of helper T cell subsets, we drew out Tregs as one of them. And so in fact, swapping from Treg to Th1 or from Th1 to Treg or something like that is in fact really deviation, or really the same kind, a similar kind of deviation. And so the idea of, if we're going to break this, basically we're making the balance not work. We're sort of shifting you, works for either deviation or suppression. We don't have to talk about those sort of separately. Also, completely forgot to mention this before. Yesterday, I did an episode of the podcast Immune, and we actually did the entire episode about Tregs and the paper that we didn't have time to discuss in this class as an homage to the Nobel Prize. So if you really want to hear more about that, it will be released in like December. It was a really fun conversation. So that would relate to suppression, which is part of this. So I'm going to talk about kind of two particularly famous examples here. And sometimes when we talk about these examples, they really help people lock this understanding of deviation into their mind. And the example, one of the famous examples I'm going to tell you about is maybe it's kind of a deviation and kind of suppression, because the idea is you're supposed to make a Treg, but you don't. You make something other than a Treg. So again, that's why it's sort of both of these things a little bit. It's also one where you can argue a lot about the antigen, and is this hypersensitivity or is it autoimmunity? I don't care, because it's the best example of this. So we're just going with it. So before I tell you about this example, I want us to think through a little bit of a scenario. We are going to talk about this scenario in a little more detail when we talk about hypersensitivity, but just thinking about it now will help us a bit. So let's imagine that I take a protein. Immunologists really love this protein called ovalbumin. It's from a chicken, and it's like everyone's favorite model antigen that we look at immune responses to. Let's imagine I inject you with ovalbumin, like in your arm. You're going to make a great T cell response to ovalbumin. CD4, CD8, you're going to make antibodies, you're going to make all this stuff. It's like the most studied antigen ever because everyone loves doing ovalbumin studies, okay? Do you think your body, notice I said body very specifically here, has ever had ovalbumin in it? Yeah. If you've eaten a chicken or if you've eaten an egg, technically ovalbumin is really big in eggs, in chicken eggs. Yeah, so you actually have already, your body has seen ovalbumin before in eating chicken or eating eggs. But you've probably eaten chicken or eaten eggs like more than once, right? And you're still doing okay. What's up with that? And again, we're going to talk about this more, but I wanted to just sort of mention it. What's up with that? So the short answer is that your GI tract is actually specialized so that usually if you see an antigen there for the first time you ever see that antigen is in your GI tract, you make Tregs instead of making other kinds of T cells to it. The idea is that like if the first time you see some antigen is in your GI tract, it's probably food. And so you should not attack that because you want to be able to eat successfully. This involves a lot of food allergy things. Again, this is going to come up more when we talk about allergy, but now that I'm on it and love talking about this, one of the reasons why people think that there might be such an increase in peanut allergies in little kids recently is because there has been an increase in use of lotions that have peanut oil. And so more and more babies are seeing peanut antigens for the first time in the skin instead of by eating them. And so one of the things that people think would be really helpful is don't use lotion with peanut oil on your baby, and also feed that baby peanut related things as early as you possibly can so that you can get that more Treg type of response to that type of antigen. Again, this becomes more of an allergy thing, but we're going to be thinking about a situation that also involves an antigen coming in through the GI tract. Yep, David.
So if you eat ovalbumin,
Mm-hmm.
and that's the first time you encounter it,
Mm-hmm.
your body doesn't.
Yeah. And then if I inject you later, you will still not have a response.
And because Tregs are the ones that are.
Yeah, because you made Tregs, and so if I inject you later with that ovalbumin, you will not make a response. So if I inject you with chicken right now, you probably will not make a huge immune response to it because you have eaten chicken in the past.
But if you inject ovalbumin into a baby who hasn't seen that in the bloodstream, then it'll.
Yeah.
Yep.
But so then they will make a very highly inflammatory response. Technically, we don't usually do this in babies. We do this in mice who have never eaten chicken before. Yeah.
So there's been studies where C-section babies are more likely to have
Mm-hmm.
certain types of food allergies
Mm-hmm.
than non-C-section babies.
Mm-hmm.
Is that just an overall thing? Because you're not eating peanuts.
Right. So the bigger answer to that is that there's some pretty good data that C-section babies start out with a different set of microbes in their body than non-C-section babies, because the place where they get the microbes from the first time they get microbes is different. And that sets up your immune system on a different sort of trajectory, is my short answer to that. There could be a much longer answer, but that's going to be my short answer. So what you should get right now is that the idea is usually if you eat an antigen, you're going to make Tregs. The issue that I want to tell you about has to do with celiac disease. And so celiac disease is tricky because there's this foreign thing, the gluten, but it chemically modifies a self protein. So is it a foreign thing or is it a self thing? Is it hypersensitivity or is it autoimmunity? Because in fact, it's gluten that's attached itself to a self protein. So so that's why I had to at least tell you these are two separate things, but whatever. So it turns out that if the first time you ever eat gluten in your life, you just eat gluten, here's dietary antigen, a dendritic cell picks up that in the GI tract, and you make a Treg, and you don't respond to gluten. But if you have the really bad luck that the first time you encounter gluten, you also have a microbe in your GI tract that you are making an infection to. In particular, you are, say, infected with this virus called reovirus. Now, your dendritic cell that picks up the gluten also picks up the the reovirus, that dendritic cell is going to make different cytokines than it would have before. Before, it would have made things to push T cells to IL-10 or to Tregs, because we don't want to kill we don't want to respond to gluten. But if you have the bad luck that dendritic cell is like doing double duty, it's responding to reovirus and it's responding to gluten at the same time, then that dendritic cell is not going to be turning on Tregs anymore. It's kind of important. We don't want to turn on Tregs in response to reovirus. You might have heard of rotavirus, which is a major diarrheal disease of children, which is sort of a virus in this family. It would be really bad if you had tolerance to rotavirus. You don't want tolerance to rotavirus. You want T cells. They're going to kill rotavirus. But the dendritic cell is doing two different things. And so the dendritic cell is going to, in fact, turn on good Th1 cells to attack rotavirus, but it's also going to be making those same cytokines, and it's going to turn on a helper T cell that is going to respond to the gluten antigen. And now you're going to get this T cell in the GI tract that is going to make an autoimmune response when you see gluten, because you had that bad luck of having reovirus infection in your GI tract the same time you first encountered gluten. You can also imagine, by and large, this is happening in really small kids, and their parents don't always have the ability to sort of go back in time and be like, oh, it was the time that you had that runny nose when you ate the bread, right? And then they show up with celiac disease a few years later. No one's sort of tying back all of those weird events multiple years before. Yep.
How does celiac develop in later stages of life?
So very likely that it took a while for the T cells to get activated and get proliferate enough. But you can also imagine, so we've got this T cell here, right? So let's imagine this T cell. So let's say the first time we saw gluten, we turned on a Treg. And then later, we got reovirus. We got rotavirus, okay? And the T cell came around. What's going to happen with that T cell when it comes around? If it got previously turned into a Treg? Yeah.
Doesn't mount an immune response because it's...
Yeah, it's going to stay a Treg. It's going to continue doing Treg things and not get you celiac disease. The problem here is if this was that T cell's first time seeing antigen, getting activated by that dendritic cell, right? Well, it is entirely possible that you could today, right now, as we are speaking, you could do VDJ recombination in your thymus to make a T cell that responds to gluten, and that T cell is naive until it gets turned into a Treg or until it gets energized. That T cell is susceptible until we turn it off. And if you are unlucky enough that it happens to go to your GI tract and it happens to find gluten and reovirus at the same time, it's going to get turned on and start leading to celiac. So you are making more cells throughout life, and they, in theory, all need to be tolerized throughout life. And so it can happen probabilistically. It sucks, but it can happen. And so you can kind of see this situation here as well. So basically all I want you to see from this is that celiac disease is complicated because in fact we have the antigen coming from the gluten, and we have the Th1 cell, and then we have damage where we actually let other antigens out with some epitope spreading. And there's modification of self-antigen, and there's a lot going on here. But the moral of the story is that we actually had this cell that was supposed to become a Treg, got turned into a Th1 because there were bad signals on its day. Yep,
So
David.
the whole confluence of events is like you have to have a naive, unenergized cell
Yep.
that binds to gluten plus reovirus, and you have to have eaten gluten and reovirus at the same time.
Yeah, it's sort of like all those things have to happen together.
And this can happen to anyone, basically.
Well, I mean, you could also wonder, like, is there something about a particular antigen, like, that has to get presented? Like, can everybody present gluten antigen? And so it might also have to be a specific HLA type. So you can see, like, it's a series of unfortunate events. Yes?
Is reovirus like super common? Because I know that like prevalence of celiac is increasing.
Reovirus is pretty common in kids. We have in a lot of situations, yes. And I am aware, this was sort of like worked out pretty well. I'm showing you some papers from 2017. There are other viruses in the GI tract that can lead to the same thing. It's just that it hasn't been described as beautifully as this story has been described. And so there are other intestinal viruses that could give you kind of the same outcome. I know of a situation where someone actually, for a bunch of reasons, is pretty sure they know which infection led to their kid's celiac, and it was not a reovirus, but it's another GI virus. So
Is this the only way you can get celiac disease?
it is the well-described way you can get celiac disease, but with the immune system, I will never say only way. There was another question, I feel like. Maybe not. Okay. We're not even at the interesting one yet, guys. The one where I was expecting all the questions. So the other place where everybody loves to talk about clonal deviation and things going on with clonal deviation has to do with Helminth infection. And so I want to tell you a little bit about Helminth infection. So when we think about helminths, there are many, these are different types of eukaryotic parasitic worms. There are many different types. They include roundworms, flukes, and tapeworms, and can lead to things like ascariasis. That was the one I showed you at the beginning of the semester with the person with the crazy huge amount of worms coming out of their GI tract. Guinea worm disease, elephantiasis, which was in my intro bio textbook as a first year student, and I was traumatized and cannot look at elephantiasis pictures ever since then. Pinworm, hookworm, river blindness, trichinosis from uncooked pork, schistosomiasis, tapeworms, lots of different situations where we have helminth infection. And helminths are pretty big, and they are best combated by a Th2 response. And so, in fact, it seems as though a big reason why Th2 responses evolved was to deal with things like helminth infection. And so you can see sort of a mast cell that might be turned on during a Th2 response. That mast cell is full of granules, and that mast cell will secrete its granules. You can see it lost them all. When it gets triggered, here you can see this in cartoon form. Granules, you release the granules. And the idea here is these granules are full of toxic stuff that will kill some extracellular parasite. So the idea is this parasite is too big, you can't phagocytose it, you can't get it inside of one of your cells, so you're going to shoot toxic bombs at it to try to kill it. And here you can see this is actually just a schistosome larva. It's not even a fully grown schistosome. And you can see here are some eosinophils, could also be mast cells, secreting toxic stuff to kill the schistosome. Here is actually an image of a schistosome larva, and those are all the little eosinophils. You can see those all sorts of little eosinophils are not eating this larva. They're just going to shoot stuff at it to try to kill that parasitic worm. And as I told you before, it turns out that if you make a Th2 response to helminth infection, you get sort of this protective response. You get rid of the helminth infection. If you make a Th1, that is going to lead to some host damage. One other thing to be aware of with regard to helminth infection is that if you are infected with a helminth, it's usually a pretty long-term infection. It might, once you have it, it's going to be for a while. We have some medicines now, so we might be able to cure you, but you're generally, before those medicines, you were kind of infected for the rest of your life. It's a long-term kind of infection. And so usually what happens if you have a helminth infection is your whole body kind of goes Th2 for a while, because you're dealing with that helminth infection for a long period of time. You're doing a bunch of Th2s. So one thing that's actually been really interesting, the sort of more recent side to this data, before I get to the classic part of this data, is that people have actually found that if you look in different parts of the world, you saw different levels of severity of COVID. And in fact, I knew some people who were in a couple of different countries who contacted me pretty early, and they're like, something's weird here. People in this country, the person who contacted me first was from Bangladesh. I was like, people in Bangladesh are not dying of this the way they're dying in other places. We don't know what's up. And it turns out what was actually happening is that people were actually infected with helminth infections. Their body was making a Th2 response. And so they were making a Th2 response to COVID instead of a Th1, because their whole body was pushed in the Th2 direction. And it turned out that if you got a Th2 response, you actually had a protective response. It was better for you. You were less likely to get really sick. And so it turned out that if you happened to be carrying a parasite when you got COVID, you actually had less severe infection. You lucked out in that front. Sad for you in other fronts, but you lucked out in that front. And people have also seen that if you happen to be carrying a helminth infection when you got a vaccine, a COVID vaccine, you actually made a worse response. So again, because you were deviating to Th2, which was not the one that was really protecting you. And so these are some really recent examples where we've been seeing deviation. And a bunch of examples of deviation. One of these papers is one of the options for the assignment that's due on Friday. But there's a deviation story everybody tells in immunology. So that's where I'm going now, is the other deviation story. And so I want us to think for a second about helminth infection in thinking about this story. So I want you to think about your life up until this moment. In your life up until this moment, how much time have you spent worrying about helminth infection? So I see a lot of head shakes of no. Why do you shake your head no? Why are you not worried about helminth infection? Yeah, Miles. Sorry, say that again? What's over here mean?
In the United States.
Yeah, so it turns out that because of a lot of sort of sanitation and types of issues, helminth infection is common in some parts of the world, but really uncommon in other parts of the world. You are very unlikely to get a helminth infection in the United States. And there are some other parts of the world where you are very likely to have helminth infection. It turns out, and so in the image on the right, in red, you can see places in the world where you can get a helminth infection. And again, this is only showing kind of high, medium, low. It's not like the most granular data ever. In blue are places where you are not likely to get helminth infection. And it was noted that if you take that graph, that map, and you look at it really closely, you can also see, so this says autoimmune. I will say that I think of this more as autoimmune and allergy. So I would also throw allergy in here. you tend to see a lot of people who have autoimmune diseases and allergy in the red areas, and you see very little autoimmunity and allergy in the blue areas. Kind of looks like there's this inverse relationship between having helminth infection and having allergies. Almost like having helminth infection is like protecting you from allergies and autoimmunity or something. And so this was an idea that some immunologists came up with called the hygiene hypothesis. There is a lot of epidemiological evidence, wrong class I teach, talking about the hygiene hypothesis and things that are statistically associated with having allergies or not. And so some of those things include that you are more likely to have an allergic disorder. So here it's calling it out as allergy. If you are from a Western country, you are less likely if you are from a developing country. You are also more likely to have allergies if you are from a small family with not very many kids. You are less likely if you are from a family with a lot of kids. You are more likely if you grow up in an urban area to have allergies. You are less likely if you grow up in a rural area. And you can also see the helminth sort of idea here. And so this is also shown, you are more likely to have allergies, asthma, etc. if you are an only child. You are less likely to have them if you have older siblings. You are also much less likely to have them if you go to daycare or if you grow up on a farm. And so all of these, actually the data is really good. And so there has been this hypothesis that has developed that basically says, as it said on this previous slide, your different types of helper T cell responses evolved to happen in balance with one another. We have basically, in Western countries, eliminated the Th2 pathogens. And so, there are no reasons why you should ever make a Th2 response, but you still should be making all the other ones. And your body doesn't know how to get rid of just Th2 and have all the other ones. You basically have now really messed up the scales. And as a result, your body has is like, I gotta make Th2 responses, but I never learned how to make Th2 responses. And so it makes messed up Th2 responses. And those Th2 responses are to pollen and dust and other types of harmless things. And so the idea is that because you don't ever train Th2 responses well in childhood, your immune system doesn't learn how to sort of make a correct Th2 response as part of this balance. It becomes poorly educated, exercised, and inexperienced in fighting Th2 infections. And as a result, it turns to fighting imaginary infections, leading to a variety of allergies. And so the idea is that because you don't see enough Th2 pathogens or enough Th2 antigens early in life, your Th2 ability gets messed up and you have excess allergies. There are ideas so it was always a joke when I was in grad school, whenever one of us would have an allergic response, someone would be like, your mom didn't let you eat enough dirt. There are actually people who are taking schistosome larvae that you saw there, killing them, and then putting them in yogurt or things like that to try to give you Th2 antigens and try to figure out ways to give you proper Th2 responses. There are some people who take a tapeworm to try to get rid of their allergies, which I'm going to say that's a no. So there is sort of this idea of the hygiene hypothesis, and these data are really good. It has kind of gotten taken to weird places in pop culture, in sort of you must be around microbes. Too much cleanliness is really bad for you. And This is very specific to Th2 responses. This is not specific to like all, you are never going to live in a sterile environment. You're never going to live completely without microbes. This is a very Th1 versus Th2 response.
The whole boy lived in a completely sterile environment without microbes.
False.
No.
He had GI microbes.
That's inside. That's internal, not external.
I'm pretty sure he probably breathed some out and otherwise excreted some.
Oh, sorry. I didn't mean to interrupt.
I will say that there is also an alternative explanation for what's going on with the hygiene hypothesis. So there are some people who kind of say that this is not a deviation thing in quite the same way when you see this type of response. These individuals say that, and this is also relatively true, that helminth infection is generally immunosuppressive. It's generally kind of pushing your immune system down. So if you think about immune systems in some of these red areas, they're just a little less strong. They're a little weaker than in some of the other areas. And so as a result, they're not going to overreact. Whereas ours are so strong that they start to overreact. Mine is so strong it starts to overreact. I personally would be in that situation. And so the idea is that perhaps in a population where a lot of people have parasites, the immune response is kind of suppressed. If we don't have parasites, you can see the curve is a little bit to the right. The immune response is stronger. And so here, it's really rare to have such a high response that you have immunopathology, whereas here, it's more common since the whole population has shifted over to the right in the response. People have also kind of modified all of this into other things because it seems like over human evolution, we probably always had parasite infection. So some people, in fact, now talk about something called the old friends hypothesis. And in general, the idea is that our immune system is evolved to be having parasite infections all the time. That sort of set point is at a place where it's assumed that you're having parasite and other types of infections all the time. And as we get rid of whatever microbes, and this could sort of broaden beyond parasites, if we get rid of whatever microbes we were supposed to evolved with, our immune systems are going to be messed up. If we get rid of whatever our old friends are, our immune response might be messed up. And so you can see this lists like all of these things in the environment as well as things in the microbiome. They all can help us sort of be regulating our immune responses correctly. And as we mess up all of those different responses, we may end up with unregulated inflammation. And so we may have chronic inflammation happening because we're not getting the regulation we should have gotten from our previous old friends. And that could lead to autoimmunity, that could lead to hypersensitivity, that could lead to inflammatory diseases, that could sort of play into all of these types of things. I think the next two slides are sort of getting at this. And so you can see diet, toxins, other things lead to chronic inflammation. That could actually deviate your immune response. And so kind of all of these things could be working together to deviate you towards a higher inflammatory state. And so maybe normally people did not make Th1s and Th17s as often, but now we don't have the regulatory responses. So we're going Th1, Th17 too much. We're seeing these responses, and that can lead to many different things. These are some of the responses or some of the disease states that have been associated with excess inflammation and deviation of the immune response away from a regulatory environment. So I will, some of this I definitely still have to talk about. So I will talk a little bit about autoimmunity on Friday. And then probably move into hypersensitivity. So I will see you on Friday. Remember, the assignment is due on Friday by 5.